Good morning there, ladies and gentlemen, boys and girls. Today we are going to talk about an integral part of all chemical reactions that need to happen in the cell, and that is something called enzymes. Enzymes are a protein. So we talked about several times that proteins are the ones that get things done, make things happen, and today is going to be a very specific example of a type or a set of type of proteins called enzymes. And there's all different kinds of enzymes out there, but we're going to talk a lot about their kind of in general their main role. So things we're going to talk about is what is an enzyme, how does it work, and then and what conditions does it work best. Just like any kind of worker, it works best in certain conditions. Just like a worker in an office, that worker doesn't work as well. Isn't as proficient, or even as me as a teacher is not as proficient when I'm working in a room that is 120 degrees. It affects my efficiency. And so that's what we're going to talk about here. Enzymes, we're talking about efficiency. All right, so let me kind of give you a little bit of a little background and a little setup here to talk a little bit about what enzymes are. All right, let me start with an analogy here of what the purpose of an enzyme is. All right, so let's say, for example, here, say I'm on the ground here and I need to move this big, heavy refrigerator. I got to move this big, heavy refrigerator across the ground, all right? All right, it's very, very big. And so right now, at this point right now, I'm probably going to be using a lot of energy to try and move it. It's me being sad. All right, trying to move it. It's very, very hard to do. It would take a lot of energy for me to move it across the ground like that. All right. In chemistry, there are these things called catalysts. Right? Catalysts are not used as part of the reaction. They're not part of the reactants, but they help make the reaction go a little bit more efficiently, in what they call speeding up the reaction. And so in this case here, this would be an example as if I was trying to move this would take a lot of energy. It would take me a long time to move this fridge all the way across the floor. But what if I took this fridge and I put it on a skateboard, a really big, heavy-duty skateboard? All right. All of a sudden, this becomes a lot easier to move. All right. This becomes a lot easier to move. And this, in this analogy here, that skateboard is what we call a catalyst. All right. A catalyst is something that speeds up a reaction or a process. It speeds it up by basically making it more efficient. I'm a lot more efficient at being able to do this. I'm using less energy to make this refrigerator move because of the catalyst here. All right. The catalyst is not now part of the refrigerator. It's just adding to it. All right. So example here I apologize. In a chemical reaction here, I have glucose or an oxygen, which makes water and carbon dioxide. In a chemical reaction, to make a chemical reaction activate, most chemical reactions need something called activation energy. All right. Activation energy is the energy you need to kind of make the reaction go. Normally, you have to put energy into a couple molecules to make it start rearranging. All right. So to make glucose and oxygen start to break down that glucose molecule, because it's very big, into smaller waters and carbon dioxides, you need about 30 ATPs worth of energy. Remember, the whole role of ATP is to break off that phosphate to release that chemical energy so that we can use it. You need about 30 ATPs to do that. All right. But if I use a catalyst, all right, if I add a catalyst to this reaction, again, something that speeds up the reaction, what it will do actually is, if I add a catalyst, it's going to reduce that 30 ATP into just using 2 ATP to get this started. That is way more efficient. If I use 30 ATP to break down every glucose, right, I'm using a lot of energy just to get this energy going. But if I can do just 2, I just saved 28 ATP to be used elsewhere. All of a sudden, things are way more efficient. All right, I can make glucose and oxygen, or I can break down glucose at a much more efficient rate, just like how I can move the fridge at a much more efficient rate because it's using less energy to do that. It requires less activation to do that. And in cells, that kind of catalyst that we do that is a protein called an enzyme. All right, it's a protein called an enzyme. And an enzyme's job is to basically, by reducing the activation energy, we are speeding up a chemical reaction and thus making that chemical reaction more efficient. Especially one like this is cellular respiration. Its job is to create more or recharge ATPs. I can recharge a net amount of ATPs, which we'll talk more about that going on. All right. So, what is an enzyme? All right. So let's talk about it specifically, because catalysts can be there's all kinds of catalysts in chemistry. All right. Biological catalysts, catalysts that we find in our cells are done by a protein called enzymes. All right. And so what a lot of times they're just called is the enzymes are biological catalysts. All right. More specifically, they are proteins. All right. And remember, biological catalyst lowers activation energy. Our catalyst lowers activation energy. That energy needed to get the reaction started. All right. So now we talked about what it is, how it kind of rolls in that chemical reaction. Now let's talk about how it actually does that. All right. And the model that is most commonly used is what we call the key and lock model. All right. So I'm going to show you what we call the key and lock model. And I'm going to show you how enzymes do this process in a very broad kind of generic sense. All right. You have to remember, especially when we talk about proteins and all biological functions, structure determines function. All right. Some enzymes they're very, very specifically to one job, to break down, this one maybe to break down glucose. But there's another one to break down lactose, the sugar found in milk, to break down sucrose. Right? And each one of them, the enzyme that does that is going to end with an ASE. So for example, the enzyme responsible for breaking down lactose, the sugar found in milk, is lactase. All right, and let me show you how it does that here. So let's say here we have our enzyme. I'm going to draw it here. It's going to have a very specific shape. All right, it's going to have a very specific shape here. This is our enzyme. All right, and it has a very specific shape for a very specific reason, because it is supposed to work with something very specific. All right, and so we're going to use kind of very general terms here. All right, let's say its job is to work with the very specific. Let's just say glucose, just to give it something right now. All right, and glucose shape, again, just kind of making this easy for this key and lock model here. All right, maybe glucose looks like. All right, let's say that's glucose. All right. Notice that its shape looks very, very similar, all right, to that enzyme, because each enzyme is specifically shaped to work with a very specific function. This one is not supposed to work one that has maybe a circle shape, right? But a one that has a square and triangle. All right. Another kind of very broad term for this right here. I picked glucose just to be very specific. This is a lot of times called the substrate. All right, it's a very general term of what an enzyme binds to. An enzyme always binds to a substrate. In this case, that substrate is glucose. All right. Now, one more thing before we kind of actually jump into this here. On my enzyme, all right, I have my substrate, which has a very specific shape. My enzyme needs to match that shape of that substrate. And it matches there at what we call here, this spot right here. This is called the active site. This whole area right here, this kind of top area, this is called the active site. This is the spot where the substrate is going to attach to the enzyme. And so every enzyme has a very specific active site, all right, to match that very specific substrate. All right, so let's say here, all right, this is called the key and lock method because it literally fits together like a key and our lock. All right, so in this case here, that enzyme is going to attach to that very specific substrate at the active site. Notice if that substrate was a different shape, it would not attach to that that active site. All right, it becomes very important there, and this is where the chemical reaction happens. All right, it might start, depending on what the actual enzyme is. In this case here, if we're doing glucose, we're trying to break apart glucose. Right, so this would kind of act as our reactants, right? Remember, reactants in a chemical reaction is what we start with. That's our reactant here. All right, in this case, we're going to be breaking it down. All right, and then with this model, we'll end up getting, all right, that enzyme is not part of the reaction. The enzyme's job is to just, in this case, break down glucose. It's just every enzyme has a very specific role. All right, so if I draw this one more time. All right, let's see, here we go. Test my art skills here. We have now our products being released. All right, so we can have our reactants, and now we have our products here. All right, so we have our substrate, our reactant attaches to the active site of the enzyme. The enzyme's job is to lower that activation energy, to break it, in this case, to break it apart and release products. Now, this could go the other way. All right, it's not always just about breaking down pieces. All right, I literally could have basically the picture going this way, where these are the reactants coming together. The enzyme's job is to fuse them together to build something bigger like glucose in photosynthesis, and thus this would be the reactant. And these would be the substrates, and then this would be the products. All right, so we can have this reaction kind of go in either way. All right, now, like in any reaction here, this is about efficiency, and so there are working environments that this enzyme works the best. All right. So there are going to be things that affect enzyme efficiency. There are three major things that affect how well the enzyme is doing its job. Its job is to lower the activation energy and break apart pieces, or to build larger molecules, all right, to make a chemical reaction more efficient. All right, there are three major things that could affect that. All right, first one is what temperature it's working at. All right, each enzyme works best at a specific range. Some work at a wide range of, in terms of Fahrenheit, this enzyme can work really well anywhere from 50 degrees to 110 degrees. Some are very, very isolated. This one works from 95 to 90. And anytime beyond that, its efficiency goes way down. By efficiency, I mean how well it works. I mean, just think about yourself, right? If you're sitting in a room that's anywhere between 65 and maybe 80 degrees, you might be uncomfortable on one of those ends, but you can still do your work. The further down that temperature goes from 50 or 60 degrees, the less work you're going to get done, probably schoolwork you're going to get done. And anything above 80 degrees, you're going to start feeling very uncomfortable, and you're probably going to get less schoolwork done. All right, it's that range here. All right, another way is based on its pH. All right, now pH. All right, is another fancy word for basically how acidic or how much acidity there is in the solution around that enzyme. That's going to help, might affect its shape, all right, and what we call it being denatured. So here's an example. Let me kind of talk a little bit about that real quick. There's a very specific pH range. pH goes from 1 to 14, 7 being neutral. All right. Say it works best in a 6. That's slightly acidic. All right. And then it's put into a 4, which is much more acidic. All right. What it will end up doing is doing something what we call denaturing. And what it does is it changes the shape. So if I put this let's say I put this enzyme in an acidity that it's not used to, all right, it will change the shape. And so maybe this enzyme turns into a circle and then a triangle this way. What I've done is I've changed the active site. All right. What I've done is I've changed the active site. And if let's say this is the same enzyme, if its job is to attach to a glucose with a square and a triangle, it's not going to work anymore. What I've done is what is called denaturing the protein. Right? Denature means to change its shape. And remember, structure determines function. So if you change its structure, you change its function. And it can't do its proper function anymore because you've changed the shape or the active site where that substrate's going to attach. All right. And the last one is pretty obvious. All right. If I have a lot of glucose to break down, I need a lot of the enzyme necessary to do that. All right. If I eat a ton of food and I have a lot of glucose starting to come into my cells, it's been broken down and now I have glucose into my cells, and I have a low amount of that enzyme to break it down, it's just not going to work as well. I'm just not going to get as much, energy from that. So the last one is basically on the concentration of enzymes. Let's say enzyme concentration. All right, so we have enzyme concentration here. All right, I know this video is getting kind of long here, one of my longer ones here, but let's just recap real quick. All right, so the purpose of a catalyst is to lower activation energy to improve the efficiency, getting more energy out of a reaction than you're putting into it, because it needs to lower the activation energy. Activation energy is the energy needed to get us started. In cells and in life, we call those biological catalysts, we call enzymes, which are a protein. And a protein that has a very specific structure and function. The big structure that we're looking for is that active site, because that determines what substrate attaches to the enzyme. Then it's either going to break down that substrate into products, or it can even take two substrates and attach them into a final larger product. Just depends on the type of reaction. It depends on the enzyme. All right, it's called the key and lock model. All right, but how well that works, how well that enzyme works, is determined by its temperature, that temperature range. All right, its pH, which might act, if it's put in the wrong pH, the wrong acidity, it might end up denaturing that protein, changing its shape. All right, and enzyme concentration, which is basically how much enzyme there is. Whew. Okay, I know it's a lot there, and there are a lot of things in there. I really, really recommend, your notebook might look just like this here, but if you're looking at your notes right now and you're like, I don't know what that word means. He wrote that and I wrote that, but I don't know what that means. Go back in the video and add yourself a little note. What does it mean to be denatured? All right. He said activation energy, and I wrote that, but I don't know what that is. All right. Either look in those textbook pages I gave you, or pause the video, go back and look at when I talked about activation energy and write your note. Your notes don't just have to look like what I put on the board, right? It should look like something that you could look back at and understand. Okay. Whew. All right. That's good for now. Thank you for watching. I know this one was a long one, but this is kind of a big topic, and I'd rather have done one big video than chop it up and do different videos. So, all right. Thank you for watching. Good luck, and may the science be with you.